Hello everyone, my name is Jillian Corredera and today I will be talking to you about my research project that I did over the summer with Miami-Dade College and with St. Thomas University. My project was about developing protocols for immunohistochemistry. So the purpose of this experiment was to study the expression of neuronal transcription factors located throughout the adult zebrafish brain before traumatic brain injury. So the procedures were pretty basic. There are only two steps to immunohistochemistry, primary and secondary staining. The staining is done in order to tag specific antigens within a sample. And the neuronal progenitor markers we were looking for were SOX2 and NeuroD1. SOX2 is a stem cell marker used to analyze the migration of stem cells from the ventricles to the injury site within the tissue. And NeuroD1 is just a premature neuronal marker. So in the figure here, in the two figures that we see here, we can see the DAPI staining of an adult zebrafish brain section. So this is just the staining without any of the neuronal markers shown in the photo. So for our results, we obtained positive SOX2 staining, which can be seen with the red circles in the images. However, we did not obtain clear positive images for NeuroD1 staining. So these negative results, they could have been due to exposure of light. What happens is that when you do the primary staining, you have to keep them in a dark drawer and make sure that there is no exposure to light. Because if not, then the images can then fade out and then it'll be very difficult for you to be able to see the staining under the microscope. So this is probably why we weren't able to observe NeuroD1. So as said before, in the figures, you can see the SOX2 markers found within the adult zebrafish brain section that we stained. So the conclusion of this study is that this can help us understand which transcription factors found in zebrafish are associated with regeneration following traumatic brain injury. So my part of the project was to do the staining before traumatic brain injury. So that way, after the traumatic brain injury, we can see if there were any changes within the transcription factors to give us light about how zebrafish are able to regenerate so quickly after traumatic brain injury or spinal cord injury. So moving forward, we want to study new neuronal transcription factors, such as the ones found here on the slide. So we want to be able to locate their immunoactivity in the adult zebrafish brain to see if these are also found and if they're also associated with the regeneration of zebrafish. So thank you. That concludes my presentation. I hope you enjoyed it.